We have polar residues here, so they can form hydrogen bonds with the water, remain soluble. But here, this is not great. In fact, we're going to see this today when we talk about sickle cell anemia. So here, right, we have this region that's hydrophobic. So we would imagine that this region is going to be on the inside, and the interior, but it's not, it's on the surface. So what would that suggest to you? How could we make this be on the inside? Yeah. No, what were you going to say before, sir? No, but oxygen is polar, right? So we wouldn't. Huh? O2 can form. We'll see how oxygen binds, but we're not going to see an oxygen binding really to a non-polar environment. What this suggests is that this region of hemoglobin should be on the inside, but it can't be. So how do we make this region of hemoglobin be on the inside? Well, take another hemoglobin, roughly, right? We just turn it 180 degrees so that that hydrophobic region is here. Well, now these two can interact together, right? And now this is kind of on the inside. So those polar residues that we find in myoglobin, which are now non-polar in hemoglobin, that suggests that that region is actually interacting with another non-polar region on something else. As a example, we'll see today, that something else is another part of hemoglobin, right? So we're going to get into way more detail on this today. Any other questions? Free homework answers? Yes, no?
Are there any other practice questions, especially for the math concepts, because it's harder and extra for practice?
So the back of the text has extra problems, so I don't assign all of the problems from the back of the textbook. You could try there. Some MCAT books also have extra problems regarding most of the stuff that we talked about. I could try to dig up a couple of old exams, try to post those. Right? Good? Alright. So moving on to chapter 3. So chapter 3 deals mostly with binding, right? So what we're talking about is a receptor or protein, enzyme, whatever, binding its substrate, sometimes called a ligand, all right, or ligand, depending on where you're from. So what's the difference between a ligand, a substrate, and a ligand, a substrate? Is a ligand typically something that binds, right, but isn't necessarily acted on. So for example, when we talk about hemoglobin, we would say that oxygen is the ligand, or the ligand, for hemoglobin. Hemoglobin and myoglobin bind oxygen. They don't do anything to that oxygen, they just move it around, okay? A substrate is something that is acted on by a protein, right? So this would be, for example, hexokinase, which binds an ATP to glucose and changes them, changing the ATP into an ADP and the glucose into glucose 6-phosphates, which is a chemical reaction here. But they all still have to bind, right? And studying how an enzyme or protein interacts with a substrate that is changed is enzymology. And that's chapter 4. We'll get to chapter 4 on Friday, right? So let's examine binding for today. And so we can describe binding as a fundamental process for anything to be acted on by a protein, for example. It has to bind to that protein, right? So nothing would get done in the cell if these macromolecules like proteins weren't able to recognize and bind to each other in a regular way, right? So let's consider a receptor and a ligand. And the reason we're going to consider receptors and ligands is because receptors and ligands do interact, but they typically don't change the nature of the ligand. So we're studying purely binding. And we can have a couple of different situations here, right? We can have, as we add more ligand, okay, the percentage of receptor bound to the ligand increases sharply, right? So as soon as we add ligand, all right, we get all the way up to 100, and then it levels off. After we've saturated the amount of receptor that we have, right, well, now we can add as much ligand as we want. We're never going to get past 100% binding. So it's an example of tight binding, right? As soon as we add an L, right, it binds, so the percentage of receptor that's bound increases linearly until we saturate the enzyme. We can also have very weak binding, right? So in this case, the interaction between R and L is very weak, between receptor and ligand is very weak. So we can keep adding L, but the percentage of receptor bound to the ligand, or the percentage of the receptor that binds the ligand, increases very slowly, right? So you can see here is at 0, here is at maybe 5% or less, right? So we have to add a lot of ligand in order to get it to bind. So this is very weak binding. What we usually find is neither very tight binding or very weak binding. Instead, what we almost always find is this intermediate binding, which is kind of in the middle. Okay, so it's neither very tight or very weak. Instead, right, we get this pseudo-linear relationship at the beginning, right, and then eventually we begin to taper off and near 100. Now, it's impossible, as we'll see mathematically here in a minute, it's impossible to have 100% binding. You can have 99.99% binding, but you simply can't have 100% binding just due to the nature of the equilibrium between the interaction of receptor and ligand, right? So this is what we usually see. And, as we'll see, right, the actual numbers, the nature of this curve is going to be dependent on the physiological concentration of the ligand. Proteins have evolved such that they respond best, right? They bind most efficiently when the concentration of their particular ligand is what it normally is in the cell, right? But we'll see what that means in a minute. All right, now most protein ligand complexes, again, exist in intermediate binding, and we can define this here L half or L50, again, depending on who you talk to. They mean the same thing. But basically, L half or L50 is the concentration of ligand at which we reach 50% fractional saturation. In other words, 50% of the receptor is bound to the ligand. Whatever concentration of ligand we have to add to get there, that's L50. And L50 is typically equal to or within the range of what we normally find in the cell. So say glucose is usually